Welcome, everybody. This is the beginning of Notes from the Lab today, an ongoing series of conversations on the past, present, and future of the BCG vaccine. My name is Denise Faustman. I'm head of the immunobiology labs here at Mass General Hospital and Harvard Medical School in Boston. Today, in this podcast series, we're exploring topics related to type 1 diabetes, the BCG vaccine, and the cytokine TNF. And my guest today is Dr. Matthias von Herrath, who joins us as VP of Novo Nordisk in the Global Chief Medical Office. Welcome so much, Matthias. We've known each other for years, and it's really a privilege to try to share these ideas, not only with the scientific community, but the really well-educated lay community. So welcome.
Likewise, Denise, I'm super happy to be here.
Yeah, these are kind of fun. And so I want people in the scientific community and the lay community to kind of understand how you got involved in the diabetes community. And what would you want to tell somebody that maybe doesn't know your science or your views in science about yourself, especially as it relates to diabetes?
So I've been at it since 1991 when I started my postdoc. And we started, of course, initially with animal models back then, and then transitioned more and more to human pathology, and then for me, for drug development, especially combination therapies. But all this is driven really by a strong passion to make a difference for the patients with type 1 diabetes. I saw this opportunity when I first came to the US. I thought we really can make a difference. And I've stayed with this ever since then, having the ups and downs of a long research journey, but that's how it goes, just like you. And you've been sticking with it for a long time too. That's just how it goes. I mean, that's the story in short.
Yeah, it's kind of a similar story. You see a signal and you go after it. Not everybody in the world may be going after that signal, but I think if you're a real scientist, you know your own data and you know what's real and what you don't know is real. And so that's why I really admire you of sticking to the data, not the dogma, and just going after the signal. So
that's a good sentence, Denise. We need to stick to the data more than ever right now, and not some type of dogmas or trends. We always have to go back to the data. Sometimes we are wrong interpreting our data. Sometimes we are right, but it always comes back to that. And we should not be afraid to do this in a circular fashion and always reach back. I think that's so important. It's often a miss now in the community since you bring it up, and I feel pretty strongly about it. That's so important. There used to be these dogmas that we always have to be right. We are 80% or 90% wrong as scientists. We should also actually, by the way, publish our negative data. That would be really good if it would be easier for the whole community, because that's the truth of the matter. 90% of the time wrong. Need a high frustration tolerance.
Yeah, I also think it's a true sign if you really believe your data, because as a scientist, you know the data better than anybody else. If you believe it, then you should be translating. And you can't just stand up your entire career with a single mouse experiment and then expect somebody else to translate. If you're betting on it, translate it, because that's the ultimate test. So I admire you for sticking to your guns on those.
Likewise.
Yeah,
likewise.
We won't go into in this podcast, but there's many times Matthias and I were at the back of the auditorium for our views. But now 20 years later, everybody believes in a CD8 cell. So it takes a long slog to get ideas out there. So everybody out there on the other side should understand that. Okay, so the next question is, what do you really do? You got this amazing title at Novo Nordisk. So tell me what happens when you wake up at 6, 7 in the morning. Is it a whole day of Zoom calls? I mean, what's your life really like with your title and your role?
So starting at the end. So I'm adverse to having whole days of Zoom calls because it doesn't give you enough time to think. We all actually need time to think, look at new data, not only our own, also from others, and put things into context. And probably in my case, from time to time take a bike ride or play some music to actually digest the data,
Yeah.
so we can think about things. So the usual day looks, because Novo Nordisk is 9 hours ahead from California.
Oh, that's
I get
true.
up, I get up pretty early, and I have a lot of phone calls early in the morning. And then the rest of the day is time to strategize and think. I think what I see as my passion and the task is I want to have a combinatorial prevention therapy at the end of the day for type 1 diabetes, and also new therapies after onset and in established disease. We owe this to the patients with type 1 diabetes, and we'll never give up. And it is not easy also in the context of a large pharma company where there are often different priorities, right? You know that's true. Mm-hmm. But the only way to lean against this is with passion for the patients and to never give up trying. So that's usually how the golden beacon in my days is orchestrated. And I try really hard to think out of the box. We had so many dogmas that type 1 diabetes is just an autoimmune disease. Now we know from the pathology that the beta cell probably contributes a lot to being recognized by the immune system. We get into this later. We had dogmas that TNF alpha is just an inflammatory cytokine, but that's much more complicated. We will dig into this in a few minutes, I'm sure. We have to question these dogmas. There's other dogmas that only FOXP3, properly methylated locus cells, are regulatory cells. There are probably many other cells that regulate immune processes and shut them down because it's so important for our immune system after it has done its deed to not do any damage. So there will be many more mechanisms and regulators. So these dogmas, we have to question them and better understand them altogether. I think that's critically important. And I'm very keen on what I thrive most on these days is interfaces. Interfaces between material sciences, be that for building best organoid platforms or spheroid platforms like we do within Sphere, or do before devices for islet implantation. Also interfaces, I had a connection with the Google folks just yesterday between AI and a better understanding how the immune system and therapies works. These interfaces I find absolutely fascinating because not only you learn yourself so much more from that, then you also get new ideas once you learn to speak the same language, which is usually the biggest problem. If you have these interfaces, you have two different disciplines with different languages. And once you start understanding each other's language, this can be absolutely fascinating. And that these are kind of my favorite things doing in a given day. In Novo Nordisk, the cross-learning between cardiovascular disease, obesity, type 1 diabetes, metabolism, these are all interlaced type of problems. And we have experts in these different areas. I find this cross-fertilization and the discussions usually the funnest parts of my days when they occur.
When it's science talk.
Yes.
Not when the applications do, or the forms do, or the employment contracts do.
Well, we spoke about this beforehand. One general problem, not only to academia, but also to large pharma, is we have to put bureaucratic and operational processes. They have their role, but they have to be put in their place so that they don't get in the way. And that has to do with hierarchies in academia and pharma, with standard operation procedures. We need them, but we don't need 50 of them probably for one process, but maybe only 2. This type of operational agility, ability to pivot, and efficacy is very dear to my heart. But that's not for this podcast, but maybe you and I write an article about this at some point.
Kind of talk. Of how to protect your day,
Exactly.
How to protect your day. So, you and I both worked on TNF. Do you want to remind everybody what TNF is and what got you excited about TNF?
Well, I came in the beginning. It was supposed to be a hallmark, what people call inflammatory cytokine. And we were interested in studying it as part of the antiviral response.
Mm-hmm.
And it is, if you have an inflammatory model, either in mice and also shown in human type 1 diabetes from the J&J trial,
Mm-hmm.
at the time of immune activation and inflammation, it's an inflammatory cytokine. It's signified also by the success story from Feldman, blocking it in arthritis ameliorates arthritis. So we all know this. It's a very big success in the arthritis market. But it very soon became much more complicated and intriguing. For example, when you ramp up a viral response, then it is clear to us that it has to contract at some point. Because if it doesn't, like you see in COVID, and it keeps on lingering, you get pathology. Early on, Oldstone and Dixon and others coined this term, immunopathology. Then the immune system, if it runs too long or amok, it damages your own body. And the TNF is already there, a molecule that helps shut down the immune response in terms of apoptotic processes. And by the way, you interrupt me if you disagree, then we can have a good discussion.
That'd be good too. Okay.
So in general, in a non-inflamed, non-diseased human individual, TNF is probably a very important, not truly homeostatic, but a tuning molecule. So that can be used to tune the immune system. So like many cytokines, it has a dual role. It depends on the disease state. And because of this dual role for many cytokines, it doesn't really make it therapeutically more complicated. We just have to be cognizant when we give it. Do we give it in early prevention? The bottom line is probably in early prevention of diabetes, inducing TNF is useful. In the middle of the islet destruction and inflammatory process, maybe not so useful. After everything is over, again, useful. Right, so we have this dualism that we need to take into account. It's not really making things so complicated in my mind, because these are not complicated to grasp disease stages in their specificity to type 1 diabetes, maybe other diseases, because we will know clearly where a patient is at in their respective journey. So that I find fascinating about TNF. And I think that's also why I always found your BCG vaccine approach so fascinating in many ways.
I mean, that's how we got going on BCG. It was from all our TNF work. And,
exactly.
Maybe it was a stupid way to go for an old drug. But, we said this has an amazing track record, okay, for safety. And everybody else is working with these things that aren't safe. We should at least try proof of principle with a safe drug first to see if we can boost TNF and see benefit in people with diabetes. And that's really what launched our 20-year program looking for BCG as immunotherapy. I mean, we obviously think maybe one of the real targets is TNFR2, but the clinical trial data is now, fairly advanced on multi-dose BCG. So that's an exciting story, and of course, is going in a totally opposite direction from everybody else. We're doing immunostimulation, everybody else is doing immunosuppression, and it's we're, as somebody said, we're always looking under a different lamppost. But it was based. I mean, for people who haven't heard the story before, TNF was discovered from BCG. They called it cacaxin because when you gave an animal model or even a human a lot of BCG, they got this cytokine response. And so it was Genentech that isolated the protein that was causing this cytokine response. And then that became known as TNF. So that association between BCG and TNF is historically kind of an important one. So we've been pleased with how BCG just keeps giving back.
It does, and it's actually quite an interesting story. People have been a bit dichotomic with all these things, also with the use of animal models. I believe animal models still can teach us good things. For example, reversal of recent onset NOD diabetes has often correlated very well with human efficacy. And the TNF story and the immune stimulation story in NOD mice is actually a very interesting one, because if you stimulate these guys, you prevent diabetes altogether. This is a very prevalent phenomenon. And when we were looking, as you know, I came out of Michael Olson's lab, and so there were viruses were very prominent, and we were looking forever for viruses that enhance autoimmunity. What we found more often than not was viruses that switched off autoimmunity. We wrote a paper on that, and some of it is in all likelihood TNF induction in the re-establishment of immune regulation to that if it's done early enough in the disease process. So this duality of the function is very evident, and I find that fascinating. And we should take probably more heat from these observations in animal models, because immune stimulation, maybe in a simplistic sense, also fills empty space up. And we have learned from the animal models that empty space, in the sense of creating space in the immune system, fosters autoimmunity. It's usually the trick when people say, oh, we can transfer autoimmunity with autoreactive cells really well. Well, only if the recipient is empty. If you take a full reception, nothing transfers, nothing. You need like an act of God to induce autoimmunity with activated T cells. That really doesn't work. So these learnings we have to take home also from animal models, right? And I find that very fascinating.
Mm-hmm. Yeah, you have to start someplace. So you always can't start in the human. So you got to start in the mouse, or an autoimmune mouse, and figure out, can you really treat them once they're fully diabetic, not just pre-diabetic. There's so much to talk about. So a lot of the world in diabetes has focused on trying to treat kids earlier and earlier, and earlier to the point of they don't have the disease yet. And most of those efforts have also been focused on immunosuppression, that's the only way we're going to do it. Do you want to think about where that field's going, of treating more kids that don't yet have the disease with drugs and aren't going to hit high on the safety list?
Well, that's right to the point, Denise. I think personally, I'm very passionate about this. I think we have to treat early. At the latest, when patients are diagnosed with 2 autoantibodies, but they are not dysglycemic yet. So you can say that they have maybe an underlying disease process, okay, because they have the autoantibodies, but clinically they're healthy. So our ethical leeway and latitude to do anything, as you correctly said, especially in younger kids, but also adults, I would argue, is very small, because we are essentially treating at that point still mostly metabolic healthy individuals who might have a variable run rate till they are diagnosed with dysglycemia, but it could be many, many years. So we do not want to give them a trade-off where they get side effects from systemic immune suppression during those years. And immunosuppression has risks. It can induce susceptibility to infections. It can induce susceptibility to cancer. We know this. It can reactivate oncogenic viruses and so forth. So in my mind, there's very few drugs that would fit that profile of early treatment. I don't think, well, let me rephrase. If we could get away immunosuppression very short, we could probably get away with it, but there's no
with
precedent.
Durable effects, with long-term.
Exactly. But there's no precedent that we can do this. These are all stay-on therapies. You look at arthritis, it's not like you take one shot of an anti-TNF and then the arthritis is gone. You need to stay.
Bye-bye. No.
Yeah, the effect is gone. And so because of that consideration, and because we cannot just have a selective reset. And maybe if we reset the whole immune system, that's really not so good either for infectious diseases. There's few things that come to mind that could work. And one is an intervention that's proven to be safe, like your BCG, and would actually, in my mind, have very important tuning aspects just in that stage. These individuals are they're immunologically full. They have small, maybe regulation defects. The BCG could correct that. There's maybe antigenic tolerance, but we haven't cracked the nut yet in terms of how to do this well. There's promising avenues, but in humans clinically, we have done it in allergy, but we have not done it routinely for diabetes and other more Th1 diseases, whatever you want to call them. And then there are some compounds that we think, this is of course from Novo Nordisk side, maybe GLP-1s, but needs to be clinically tested. There's some evidence they can be anti-inflammatory and beta cell preserving. That's published, but this needs to be tested in a trial, obviously, before anyone can recommend anything to patients, and maybe other beta cell protective compounds. That's the drug classes I can see. And you can take vitamin D, but you can take vitamin D anyways. There's good studies about the benefits of vitamin D.
There's other reasons you should take it. Yeah.
Exactly. There's many reasons you should take it. But that's, of course, not, I don't think we need a trial for that. We can just take it because there's so many reasons for taking vitamin D. But that's the compounds that we would have to trial out ideally are sort of in that range, I would think. Yeah, and I found it particularly fascinating. I don't know whether you want to say anything about this, but when I followed your story, Denise, I didn't know that before, but there are different BCG strains with different effects. This came for me. I just thought this is all the same stuff. And then I learned from you and your teams, and that is entirely different, right?
You're not the only one. During the COVID pandemic, everybody ran out to test BCG in mice and BCG in humans. And, the groups up in Canada testing Tice BCG in mouse models couldn't prevent COVID. And then all the groups at Stanford goes, well, you're using the wrong strain. And we're able to then use correct strains and show it protect from COVID. So the story keeps repeating itself because people don't understand once you culture a bacterium for 125 years, the genes kind of change in significant ways. There's a lot of evolution going on in the culture dish for 125 years. So, the lessons we learned in bladder cancer, the guys doing bladder cancer have known for years that when Sanofi was on the market with their Pasteur Canot strain, it had much higher efficacy than the Merck strain for Tice. So you ask any, and there were randomized trials, but you ask any self-respecting urologist, they go, no, we want the Sanofi one. So there, clinically as well as basic science-wise, there are big differences that change those slopes for sure. So I don't want to go over time because, I'm going to go over my allotted time for notes from the lab. But if you had a magic ball, what do you think is going to happen in the next 2 to 4 years in type 1 diabetes?
So I'm actually, I'm a glass half full type of a guy. I'm optimistic. I think with all the new activities, notably by Sanofi and by Lilly, hoping personally that Novo Nordisk is going to join the party eventually, we will make a difference in prevention of type 1 diabetes. That will be very good, and at earlier and earlier stages with the right choices of the drugs. What we need to overcome, because we cannot run 59 million prevention trials with single drugs and probably need a combo therapy at the end with a beta cell and an immune component, we need a good prioritization algorithm, maybe with AI. That would be very good, because if we're not going to be able to run so many trials, how difficult it is to run trials. I'm also optimistic about the recent onset space. I think with the right combination therapy, that would again be an immune modulator and something that provides maybe immune tuning and something for the beta cell. The endpoint ideally should be to have patients to be able in a standardized fashion to forego mealtime insulins. That would get us around the whole C-peptide discussion and would get us right to the home run, because from a regulatory perspective, from a patient perspective, it's a non-arguable benefit where everybody says, well, that's good. They have a much easier life with many less decisions they have to make.
Yeah.
And then, of course, in established diabetes, I'm having great hopes for your approaches. And we should see also at some point some advances in islet replacement or stem cell-derived islet replacement. I think these will fully depend on how we are able to tackle immune suppression or immune evasion. And that's a similar problem like in early prevention. We need to prioritize this, because one can easily imagine that we're not going to have worldwide 500 islet transplant trials. These are complicated trials to run. You need to have, at the end, enough patients to draw a conclusion. So we need to have a clear priority algorithms, which immunomodulatory drugs we can replace or completely forego, in conjunction with what immune evasive properties are bestowed upon beta cells or islets, depending on genetic modification or encapsulation devices, if that field moves ahead. So that's my vision. It's actually exciting for many stages. It is more exciting for sure than 10, 20 years ago, where things looked much more bleaker. The approval of anti-CD3 has certainly lent the field a lot of energy. I think that's, from my perspective, it's a very good start. Can it be improved upon? I'm sure, also with combinations, but it's a start. So now we can see that we can build on this and make life for patients better. So I'm really excited. This is a good time altogether.
Well, I'm glad you're working on it.
Well, you are too. This is so important. It's so important.
Well, I just want to thank you for taking your time today to join this podcast, Notes from the Lab, and of course about diabetes and the hope and the vision for where we're going. And everybody has their dream, and the idea is pursue the dream and test it in humans. So for all our listeners today, if you want more information about this podcast series and our BCG clinical trials, please go to www.faustmanlab.org. And we hope we've added a little bit of knowledge and excitement about what's really happening in the ditches and how people are thinking individually. So thank you so much for joining us again. Appreciate it. Okay.
My great pleasure.